So dear students, continuing with the development of blastocyst. During the beginning of the second week, the blastocyst is partially implanted, means it is partially embedded into endometrial stroma of uterus. So first we shall see the changes happening in the embryoblast. Here, during second week, the most significant development is this embryoblast cells, they differentiates into two cell layered structure which is called as bilaminar germ disc. So it is two layer bilaminar germ disc. The two layers are epiblast and hypoblast. So the first structure which forms here, the inner cell mass is here, and we can see a line of cells formed here which is quoted as endoderm. Actually we have to write as hypoblast cells, which is a primitive endoderm actually. These are hypoblast cells. And dorsal cluster of cells. So this dorsal cluster of cells, they get organized into a columnar cells which are called as epiblast cells, which is otherwise called primitive ectoderm, or normally we say as epiblast cells. So embryoblast cells differentiates into epiblast and hypoblast. So the inner cell mass embryoblast of blastocyst stage gives a ventral roofing of the blastocyst. So hypoblast cells, you can see they form a roof. So this is the cavity here, which is yolk sac cavity. So the blastocyst cavity is now termed as yolk sac cavity, which contains uterine fluid and provides nutrition to the developing conceptus till the placenta develops. So the embryoblast cells are arranged in the dorsal side. If we see, they arrange in a long columnar cells. So they get organized as a long columnar dorsal layer called as epiblast or primitive ectoderm. So both the layers together form a flat ovoid shaped disc, bilaminar germ disc. Okay. Now the epiblast cells, which are the columnar cells present dorsal to the hypoblast cells, there we can see a cavity starts developing dorsal to the epiblast cells, and this cavity slowly the epiblast cells, they starts differentiating, they starts growing, and they line the cavity. So once they line the cavity, this cavity is called as amniotic cavity, and the cells which are lining are called as amniogenic cells, which are derived from the epiblast cells. So these cells organize to form a thin membrane later, which is called as amnion or amniogenic layer. The cavity within it is the amniotic cavity. So next about the hypoblast cells. Hypoblast cells, here they are the cuboidal cells, and the cuboidal cells which roof the blastocyst cavity, which turns to form 𝐒𝐵𝐽𝐿omic cavity. And now this blastocyst cavity is called as 𝗪𝐴𝐺𝐿omic cavity, which later turns to yolk sac cavity. So how it turns, we will see now. These cells proliferate. Now the hypoblast cells, they start dividing, they proliferate and migrate, forming the inner lining of the 𝐒𝐵𝐽𝐿omic membrane. So they migrate like this and they form the inner lining along the 𝐒𝐵𝐽𝐿omic membrane. So now this cavity, which is called his primary yolk sac cavity. Now the 𝐒𝐵𝐽𝐿omic cavity turns to form what? Primary yolk sac cavity, which is in many of the textbooks it is now yolk sac is termed as umbilical vesicle. So it is called as primitive umbilical vesicle, or easy to say primary yolk sac cavity. Now look at this picture. We can see the amniotic cavity primary yolk sac cavity, which is primitive umbilical vesicle, and bilaminar germ disc is formed where ectoderm endoderm is formed. So the epiblast and hypoblast cells form a two-layered structure called as bilaminar germ disc. So the bilaminar germ disc lies between the amniotic cavity and yolk sac cavity. The epiblast and hypoblast fuse at a point. So if we see the bilaminar germ disc, they fuse at one point, which is called as precaudal plate, which marks the future site of mouth and gives cranial caudal orientation to conceptus. So now we can say the conceptus, this is the cranial end where we see the formation of precaudal plate. So this is the cranial side. Now the other side is the caudal side of the developing conceptus. Let's see the further development of extraembryonic mesoderm. So the peripheral cells of the primary umbilical vesicle, that is the primitive yolk sac, the peripheral cells forms a layer of loosely connective tissue which is called as extraembryonic mesoderm. So here this is the extraembryonic mesoderm. This is the primary yolk sac now turned to form secondary in this picture. So these cells, they are dividing to form extraembryonic mesoderm. And slowly within the extraembryonic mesoderm, vacuoles start appearing. Vacuole means gaps. So later these vacuoles, they all join to form a cavity. So the cavity surrounds the yolk sac and the amniotic cavity except at the connecting stalk. So this cavity is the extraembryonic coelom. The extraembryonic coelom, which is otherwise called as chorionic cavity, also called as chorionic sac. So the wall of the chorionic cavity is called as chorion. So the chorion consists of three components. One is extraembryonic mesoderm, and that is somatopleuric extraembryonic mesoderm. That is how we have to say it. Then trophoblast, we know it is differentiated into two layers, inner cytotrophoblast and outer syncytiotrophoblast. So the three components of chorion are cytotrophoblast, syncytiotrophoblast together, and somatopleuric extraembryonic mesoderm. So that these are the three components of chorion. The fluid-filled cavity surrounds the amnion and the umbilical vesicle, that is the primary yolk sac, except where they are attached to the chorion. Okay, so except where they are attached to the chorion, this cavity extends. That is the extraembryonic coelom where the connecting stalk develops. Except where they are attached to the chorion, so somato-mesoderm of the trophoblast. Here we can see the splanchnopleuric and somatopleuric, they form a common component at the connecting stalk. Extraembryonic coelom splits into two parts. Again we know what are those two parts. Now we will tell. One is somatopleuric extraembryonic mesoderm. Somatopleuric is present along the trophoblast forming the chorion. The other is the splanchnopleuric extraembryonic mesoderm, which lines the yolk sac cavity. So forming into a secondary yolk sac cavity or secondary umbilical vesicle. So somatopleuric, somato means body wall. So this somatopleuric mesoderm forms the inner lining of the body wall, like it forms the parietal pleura, parietal pericardium, parietal peritoneum, all parietes. So the parietal are from the somatopleuric mesoderm. Splanchnic, splanchnopleuric. So splanchnic means viscera. So the layer with the viscera, that is splanchnopleuric mesoderm, forms the lining of the visceral organs which present around the yolk sac. That is a visceral pleura, visceral pericardium, all these are derived from the splanchnopleuric mesoderm. Talking about the yolk sac is otherwise called as umbilical vesicle, and it is a membranous sac attached to the embryo, now we know, and it is formed by the cells of hypoblast adjacent to the embryonic disc. So all this we saw it now. The first element seen within the gestational sac during pregnancy. So this yolk sac is the first element appears during pregnancy. So when the woman wants to confirm her pregnancy, under ultrasound guidance, the doctor checks for the gestational sac, that is the yolk sac. Usually at 3 days of gestation itself it appears. So this yolk sac, it is situated on the ventral side of the embryo and it is lined by the hypoblast cells, which are now otherwise called as extraembryonic endoderm, and outside which layer there will be a extraembryonic mesenchyme, that is splanchnopluric layer of extraembryonic mesoderm or mesenchyme forms. So the yolk sac develops from the cavity of the blastocyst and passes through 3 stages. First stage is the primary yolk sac cavity. So this is the primary yolk sac cavity. It is a vesicle which develops during the second week of development, where the blastocyst cavity gets converted into primary yolk sac, and as it gets lined by a flattened cells which are derived from the hypoblast. So the flattened cells which are derived from the hypoblast called as 𝐒𝐬𝐴𝐺 membrane. So once this is lined, it is no more called as blastocele or blastocyst cavity. It is called as primary yolk sac cavity. And as it gets lined by the flattened cells derived from the extraembryonic endoderm derived from the hypoblast cells forming a 𝐒𝐬𝐴𝐺 membrane, it is called as primary yolk sac cavity. And this lining formed by flattened cells called 𝐒𝐬𝐴𝐺 membrane, and which is attached to the undersurface of the hypoblast by embryonic disc, and it is also known as 𝐒𝐼𝐺 cavity. So this primary yolk sac, it is otherwise also called 𝐒𝐼𝐺 cavity. Now this primary yolk sac turns to secondary yolk sac, that is because of formation of extraembryonic coelom. So primary yolk sac is formed when extraembryonic coelom develops in the extraembryonic mesoderm. So as the extraembryonic coelom forms the primary yolk sac, what happens? The umbilical vesicle or the primary yolk sac reduces in its size. So the part of the yolk sac gets pinched off into the extraembryonic coelom, and this reduced umbilical vesicle size is called as secondary umbilical vesicle or secondary yolk sac. So what happens? I'll tell again. This secondary yolk sac, how it forms? The primary yolk sac, initially it is little larger in size. Because of the formation of extraembryonic coelom, what happens? The part of the yolk sac is pinched off from the mother yolk sac. The part of the yolk sac is pinched off so that all by the extraembryonic coelom, it gets absorbed into that extraembryonic coelom. So this reduced umbilical vesicle, which is surrounded by splanchnopluric extraembryonic mesoderm, is called as secondary yolk sac. So primary yolk sac turns to form secondary yolk sac. So it is the remnant of the primary yolk sac becomes smaller and it is lined by the cuboidal cells which are derived from the hypoblast cells. The third stage of the yolk sac is the definitive yolk sac. So here we can see the bilaminar membrane, epiblast cells, the yellow are the hypoblast cells, and we can see the hypoblast cells differentiate to form 𝐒𝐬𝐴𝐺 membrane. So this is a primary yolk sac. You can see the vacuoles appearing in the extraembryonic mesoderm called as extraembryonic coelom. Okay. Now the extraembryonic coelom forms divides the extraembryonic mesoderm into splanchnopluric. The other is the somatopluric. Now the part of the yolk sac, it reduces its size to form a secondary yolk sac because the part of the yolk sac is pinched off into the extracoelomic cavity. Now this secondary yolk sac will turn to form a definitive yolk sac during 4th week of development. Means the part of the yolk sac gets surrounded by the endoderm. So till now we haven't seen the 3rd week development. So the 3rd week there will be ectoderm, endoderm, mesoderm. So the endoderm surrounds around a part of the yolk sac which is called as definitive yolk sac, and it is incorporated into the embryo as the gut, and the remnant of the yolk sac is called as final, that is the definitive yolk sac, and it communicates with the midgut via vitellointestinal duct. So these things we will see during later development in definitive yolk sac. So one thing as of now you should know that yolk sac we have seen in 3 stages. One is the primary yolk sac. So primary yolk sac means it is lined by the 𝐒𝐻𝐼𝐴's membrane alone, which is derived from the hypoblast cells. Second stage is the secondary yolk sac. Secondary yolk sac along with the 𝐻𝐻𝐼𝐴's membrane we have got surrounded by extraembryonic mesoderm. So extraembryonic mesoderm, there will be a coelom which develops called as extraembryonic coelom, which differentiates this mesoderm into splanchnopluric extraembryonic mesoderm and somatopleuric. Splanchnopluric extraembryonic mesoderm along with the 𝐻𝐻𝐼𝐴's membrane is the secondary yolk sac. And one more change it happens in the secondary yolk sac is part of the yolk sac is pinched off into the extraembryonic coelom, reducing the size of the yolk sac. So that is the secondary yolk sac. Final stage is the definitive yolk sac. So the definitive yolk sac, that is the 3rd stage, where the yolk sac is surrounded by the endoderm. Then it is called definitive yolk sac, and it gets incorporated into the developing embryo as the primitive gut, and it communicates with the outside yolk sac by vitellointestinal duct. So these things we will see again during 4th week of development. Let's see about changes happening in the trophoblast. So all this time we observed over the embryoblast cells and changes in the yolk sac and extraembryonic mesoderm. All this we observe now. So next we shall discuss about the changes happening in the trophoblast. So at beginning, the 2nd week of trophoblast, the cells differentiate into inner cytotrophoblast. So these cells, this cellular layer is the cytotrophoblast, and the outer layer is the syncytiotrophoblast. So what happens? What is the difference between cyto and syncytio? We already discussed, right? That cytotrophoblast, they are actively dividing cells which are present in the form of cells along with the cell membrane. So they are mitotically active cellular layer. Whereas the syncytiotrophoblast, it is an outer layer where the cell membranes fuse, forming a protoplasmic mass, and which does not divide. And syncytiotrophoblast subsequently erodes the endometrium of the uterus and completes the implantation. So by day 9, lacuna develop in the syncytiotrophoblast. So you can see here, you can see these gaps which develop within the syncytiotrophoblast, which are called as lacunae. So these gaps are called as lacunae, singularly lacuna. So by day 9, this lacuna will develop in the syncytiotrophoblast. And syncytiotrophoblast erodes the maternal blood vessels. Now this syncytiotrophoblast, it is having maternal blood vessels here. So this syncytiotrophoblast erodes the maternal blood vessels which pour their blood into this lacunae. So the lacunae get filled with the maternal blood. Right. So the syncytiotrophoblast erodes the maternal blood vessels, that is uterine vessels present in the uterus, and now the maternal blood enters the lacunae of the syncytiotrophoblast. So this established first uteroplacental circulation by the end of 2nd week of development. So the syncytiotrophoblast produces human chorionic gonadotropin, that is called 𝐻CG hormone. So this syncytiotrophoblast produces, I am writing short form, 𝐻CG, which is called human chorionic gonadotropin hormone. So primary chorionic villi are formed with this human chorionic gonadotropin hormone. So simultaneously what happens? The cells of the cytotrophoblast forms a thickening and at the triangular interval. So you can see here the cytotrophoblast, it is not uniform thickness all around the developing embryo, and where we can see the thickenings at each level. So this cytotrophoblast starts thickening, invading into the syncytiotrophoblast. So what happens? This thickening further invades and they proliferate and into the as a cellular columns, penetrating into the surrounding syncytium. So for example, so this is the syncytial layer, this is the cytotrophoblastic cell layer. So these cells will proliferate into the syncytial layer like columns, and now here surrounded by what? Lacunae. So this is the primary villi. It is called primary villi developing in the trophoblast, that is in future it forms placenta. This is the lacuna which we are seeing here. This is what the lacuna is, present within the syncytiotrophoblast, filled with whose blood? Maternal blood, that is uterine arterial blood. So uterine arteries are here within the endometrium. So the uterine arteries pour their blood. So syncytiotrophoblast, so the lacunae are filled with the maternal blood, and this columns of cell is the primary villi which is formed by the cytotrophoblast. Okay, so these are called primary chorionic villi. And during second week of development, we know that implantation completes. That is, the pre-embryonic period is completed by the end of second week of development. So embryonic week starts with the third week of development. So till the second week, we can say it as conceptus. So by the end of the second week of implantation is completed, and the surface defect is completely healed. So you can see here, this is the surface defect which completely heals. So implantation begins in the end of the first week, that is approximately on 5th or 6th day after ovulation, and this implantation completes during second week, that is around 10th or 11th day the implantation gets completed. So this implantation begins with the disappearance of zona pellucida, which is called as zonal hatching. So this occurs during the invasion of the endometrium by the trophoblastic cells with the help of proteolytic enzymes. So the blastocyst implants within the endometrium, called as interstitial implantation, which we discussed in the previous session during first week of development itself. That implantation which is occurring in the human is interstitial, because the concept is here, it is completely into the endometrium of the uterus. It is completely within the wall of the uterus. So that is called interstitial implantation. And normal site of implantation, we know it is the superior part, fundus posterior wall of the uterus. So implantation outside the uterine cavity leads to ectopic pregnancies, and most common ectopic implantation is in the uterine tube or fallopian tube, causing tubal pregnancies. The other ectopic pregnancy may result due to implantation in recto-uterine pouch, or the implantation may occur in the ovary or in the mesentery. So all these are ectopic pregnancies. And the summary of the second week of development is, one is trophoblast differentiates into cytotrophoblast, syncytiotrophoblast. Embryoblast differentiates into bilaminar germ disc, that is 2 layers, epiblast and hypoblast. And one more change which we observed is extraembryonic mesoderm is formed, where it splits into somatopleuric mesoderm and splanchnopluric extraembryonic mesoderm by formation of extraembryonic coelom. And there are already 2 cavities around the conceptus, that is the amniotic cavity and the yolk sac cavity. Amniotic cavity is related to epiblast cells. Yolk sac cavity, it is roofed by the hypoblast cells. So this completes the second week of development. Thank you.